All right, let's go ahead and get started. So first, I'm just going to finish up the bone tissue chapter. We don't have that much to actually go through, which is nice. We're almost at the end. So what I'm going to talk about today with bone tissue is mostly this idea of homeostasis in minerals between bone and blood. So that's what I'm going to focus on. So I've talked about how bone tissue will basically be dissolved by certain cells and then built by other bone cells, right? So I'm going to talk about some of the physiology, the physiological aspects of that and why it matters. And then that'll wrap up bone tissue. On Friday, we'll get into the skeleton. Okay. Would you mind closing that door or like just a little bit? She's really loud. Thank you. Ah, no. That's not what I wanted. Okay. All right. So we talked about, I think I ended here on Monday. I talked about, bone remodeling essentially. So now the physiology of bone. I've said this before, I believe in here, but we typically think of bones as sort of these static pieces of our body that don't really change. Maybe we lose some bone tissue over time as we get older, but even that we don't really think about all that much. We think they always stay the same. And in reality, they're sort of constantly changing. So if you remember on Monday, I mentioned that you basically have a whole new skeleton every 10 years. So every decade, all of your bone tissue that you had 10 years ago has been replaced by all new bone tissue. So that is due to this metabolic process of bone being dissolved and then basically rebuilt. So that's what I'm going to talk about today, particularly in relation to how that affects the exchange of minerals and ions between the bone and tissue fluid. And in particular, I'm talking about blood. So movement of minerals from your bone into your blood, and then vice versa from your blood into your bones. So that's basically the end of this chapter that we'll wrap up with. Mineralization and resorption are the terms for each of those. And I will define them on the next slide, so you have a definition of them. So mineralization is also called mineral deposition. That's where minerals are deposited into the bone tissue. So minerals are moving from typically the blood into the bone. This involves, I'll focus mostly on calcium, but it involves calcium and phosphate to a large degree, along with some other smaller ions. These materials, these minerals are being essentially deposited on collagen fibers in the bone matrix. So you have that kind of, the hardened bone matrix, but it's hardened because you have this calcium attaching to these collagen fibers. That's what's creating that hydroxyapatite. So if you don't remember that term, definitely go back and look at what that is. But hydroxyapatite makes up the inorganic or ceramic portion of the matrix of bone tissue. And the matrix is the stuff surrounding the cells, essentially, the bone cells. So those collagen fibers become encrusted with minerals, calcium and phosphate mostly, and then that hardens and you get that hard bone matrix. That is mineralization kind of in a nutshell. So minerals are moving from the blood into the bone. Resorption is the opposite, where bone is dissolved. That is going to release minerals into the blood. This is the cell type that is responsible for that, the osteoclasts, clasts with a C. Those are the bone dissolving cells. So resorption is pretty much all due to the activity of osteoclasts. Oh, sorry guys on Zoom, I didn't move forward there. So mineralization and resorption, it's kind of this constant back and forth between these minerals moving from the blood into the bone and then the bone back into the blood. Okay. I'm going to focus, like I said, heavily on calcium. I'm not really going to talk much about phosphate, well, at all about phosphate beyond this slide. But the movement of calcium and phosphate from bones into blood, that resorption process, and the mineralization process, so that kind of back and forth is really important, basically to allow for the correct levels of calcium and phosphate in your blood, as well as to allow for the functioning different physiological functions and production of different really important molecules in your body. So phosphate in particular is a component of a lot of different extremely important molecules in your body, including DNA and RNA. ATP, which is the energy, the fuel source for cells. It has 3 phosphates on it, triphosphate. Phospholipids, obviously phosphates in those make up the cell membrane of every single cell in your body. And they're also, phosphate is also important in pH buffering. So we need phosphate in our blood in order for it to get to cells to make all of these molecules. The skeleton kind of acts as a storage facility for it. So it's a good kind of long-term storage. It can be released as needed. Same idea behind calcium. So calcium is very important in neuron communication and muscle contraction. So we'll talk about those when we get into the nervous and muscle tissue chapters. And it's also important in blood clotting. So yeah, like I said, these minerals are essentially deposited in the skeleton to be stored. So it's a great storage facility for calcium and phosphate, along with a lot of other stuff. And then they're taken out when needed. So when they're needed for neuron communication, muscle contraction, to make DNA, to make phospholipids, all that stuff, they can be taken out of the skeleton by basically dissolving the bone and used for other purposes. So you can think of the skeleton as kind of a storage facility for these molecules. Okay, so yeah, from here on, I'm going to focus on calcium. We're going to talk about the homeostasis of calcium between blood and bone. So how much calcium is in your blood is very important. You don't want too much and you don't want too little. You don't need to know this number, but just to give you an idea, kind of a baseline for what's normal in blood, the normal calcium concentration in blood is typically around 10 milligrams per deciliter. So going above that or below that causes some pretty significant physiological issues depending on how far away from that baseline a person is. It's kind of not good if you're too low or too high on the blood calcium concentration scale. So hypocalcemia, if hypo means low, so too little calcium in your blood. So this is referring to calcium in the blood. We're not ignoring the skeleton, but we're talking about how much calcium is flowing around in your veins. If there's not enough calcium in your blood, that's hypocalcemia. This can cause issues with muscle contractions, your heart probably being the most important muscle. So heart contractions can be thrown off, which can affect circulation of blood throughout your body, obviously, and it can also negatively affect nerve function. So too little calcium, not a good thing. And as you can see and have probably guessed, too much calcium is also not good. We typically don't think of having like too much calcium in our bodies. And you can't, I guess you could ingest too much calcium if you go way overboard, but again, most of it is stored in the bones. If for some reason, there's kind of the storage tank is filled up in your skeleton and there's too much calcium getting released into your blood, this can cause a lot of issues as well, including coma. So if you have too much calcium in your blood, you can actually go into a coma. As crazy as that sounds, calcium is typically a good thing, but we don't want too much of it in our blood. So it can lead to an underactive nervous system. So again, calcium will come back up when we talk about nervous system function. So when nerves basically communicate with each other, they need a flow of calcium. If there's too much of that, it can lead to an underactive nervous system, which will typically lead to lethargy, which is just being tired, loss of appetite, and then, yeah, in really extreme circumstances, coma. So it is really important that we keep our blood calcium levels, and not that you have much control over it, but it's important that your body keeps the blood calcium levels kind of within this sweet spot, right around 10 milligrams per deciliter. This homeostasis of calcium is going to depend on a couple of different factors. First, how much are you ingesting and excreting from a dietary standpoint? So dietary intake and excretion obviously will affect how much calcium is in your blood. I'm not really going to focus too much on that. The excretion part will come up a little bit in just a minute. But the biggest part is the exchange between the bone tissue and blood. So how much calcium is moving from bone tissue into blood and vice versa. So those are the two factors that are going to determine whether your blood calcium levels are balanced or unbalanced. And this is actually regulated by a few different hormones. Two of them sound really similar. One of them sounds totally different. The top two here, calcitriol and parathyroid hormone, are both going to raise the blood calcium level. So annoyingly, the two that sound similar, calcitriol and calcitonin, have an opposite effect on blood calcium levels. So it gets a little bit confusing in here. I'm going to describe kind of what each of these do. I have a slide for each of them. So the next few slides, I'll be talking about what these three hormones do, but kind of helps me to group these together. Calcitriol and parathyroid hormone, those are going to raise the amount of calcium in your blood. Calcitonin is going to decrease the amount of calcium in your blood. All right, so we've got calcitriol, parathyroid hormone, or PTH, and calcitonin. I'm going to talk about each of those pretty briefly here and talk about kind of how they work and their physiological function. The first one, calcitriol. You may remember this word from the integumentary system chapter. This is sort of the precursor to vitamin D. So this is used to make vitamin D eventually. We have it in our bodies once we absorb some sunlight. The process occurs where the kidneys and the liver will continue the conversion of that into what we know as vitamin D. So it's not just used in calcium homeostasis. This is going to, like I said, raise the blood calcium level. And it does that by increasing the absorption of calcium by the small intestine. So all it does when it's released, when calcitriol is released, it will encourage the small intestine, rather than excreting calcium that's going through, it'll encourage it to reabsorb it. So once it reabsorbs it, it can get back into the blood, and then that will raise the blood calcium level. So that's how calcitriol functions. Pretty simple. Increases absorption in the small intestine. Fairly straightforward. And then we have the other one that raises the blood calcium level, PTH. PTH is released by the parathyroid glands. Those are up near the thyroid gland. We haven't talked a whole lot about glands, but sort of near the brain. And the thyroid gland will come up when I talk about the next hormone. So it's really easy to get some of these confused. They have a lot of similarities. So parathyroid hormone is released by the parathyroid glands. If the blood calcium level is too low, that sends a signal to the parathyroid glands to release some PTH. That's going to raise the blood calcium level through a few different pathways. So first, it will stimulate those cells that dissolve bone, osteoclasts. So that bone will be dissolved, releasing calcium from it, and that calcium will go into the blood. That's going to bring the blood calcium level up. It also promotes absorption of calcium by the kidneys. So similar to calcitriol, how it promotes absorption in the small intestine, PTH is going to promote absorption in the kidneys. So less calcium is lost through excretion in urine. So similar idea, just slightly different, a different organ. And this is where PTH and calcitriol actually not necessarily work together, but PTH will actually enhance calcitriol synthesis. So calcitriol is the one I just talked about that also raises the blood calcium level. So PTH will enhance the production of calcitriol in the kidneys. That will obviously lead to the reabsorption, increased absorption of calcium in the small intestine. So not only does PTH kind of do its own thing, but it enhances that calcitriol synthesis, which also helps. And over here, you can see the kind of a diagram of the different pathways in which PTH can work. So here it's dissolving bone. It's going to release calcium. Here it will, well, we'll go down here. It'll promote the reabsorption of calcium in the kidneys, and it will also promote the synthesis of calcitriol. That calcitriol will then promote absorption in the small intestine. So for you guys on Zoom, if you didn't see that, bone is being dissolved by osteoclasts. That's going to release calcium into the blood. The kidneys will reabsorb more calcium than they would normally. And also calcitriol will be synthesized. And that calcitriol will then enhance absorption in the small intestine. So there's a few different pathways by which PTH actually raises the blood calcium level. All right. So the last one that lowers the blood calcium level, calcitonin. This is secreted by the thyroid gland. So PTH is parathyroid gland. Calcitonin is secreted by the thyroid gland. So all very close to each other. This happens if blood calcium levels are too high. When this is released, it'll bring the blood calcium levels down in a couple different ways. In two, I think, pretty obvious ways, actually, if we think back to how those different types of bone cells function. So it will inhibit osteoclasts. So we don't want bone being dissolved. If there's too much calcium in the blood already, dissolving more bone and releasing more calcium is not what we want, right? So calcitonin is going to inhibit the activity of those osteoclasts or bone dissolving cells. And then at the same time, it'll stimulate the cells that actually create new bone or bone matrix, specifically the osteoblasts. So it'll inhibit osteoclasts, which will inhibit bone resorption or dissolution. And it'll stimulate the osteoblasts with a B, which will encourage deposition of calcium back into the bones, taking it out of the blood, which will, yeah, bring that blood calcium level back down. All right, so you want to keep those three hormones kind of separate in your brain and understand the pathway by which each of them affects blood calcium levels, and know obviously which direction it will push blood calcium levels up or down. And here is a not excellent chart. It's a little bit confusing, but if you stop and kind of take a minute to look at it, I think it's kind of helpful. It just puts it all sort of in perspective. So I'm going to run through this real quick. So what we have here is, this is where we're happy. We have about 10 milligrams per deciliter of calcium, right in that kind of happy medium here. If we go too high, so we go above 10 milligrams per deciliter of calcium in our blood, that is going to stimulate the thyroid gland to release calcitonin. So for you guys on Zoom, here we're going above the ideal blood calcium concentration. That's going to stimulate the thyroid gland to release calcitonin. That means the osteoclasts are going to be inhibited. So the osteoclasts are the ones that dissolve bone. We don't want bone being dissolved because we don't want any more calcium released. So osteoclasts are inhibited. And then that's gonna, which I didn't really talk about, is gonna lead to reabsorption in the kidneys. So that calcium will be, it'll be the decreased reabsorption of calcium in the kidneys. So the blood level, calcium goes down. Calcium blood levels go down in the end. So calcitonin is the only one that is going to bring down, out of these three, bring down blood calcium levels. On the other side, you have, if we're on the lower side, so we don't have enough blood calcium, that's going to stimulate the parathyroid gland, PTH, or to release PTH. So we're down here now. We need more calcium in our blood. That will stimulate the osteoclasts. That's going to release calcium from the bone. It'll dissolve the bone. Calcium will come out and go into our blood. It'll also enhance reabsorption of calcium in the kidneys. And then this graphic doesn't actually mention calcitriol, but calcitriol would kind of come in over here, where the release of PTH is going to stimulate the synthesis of calcitriol and increase the reabsorption of calcium in the intestines. So not only in the kidneys, also the intestines, or small intestine. And then that will bump the blood calcium level up, hopefully. Okay, so I'll definitely ask you about this calcium homeostasis, the pathways and stuff on the next exam. And it can be difficult and very confusing as you're trying to figure out what's going on. So I've found a good way to kind of study this material or create study material for it is to sort of create your own flow chart. So you have one option of blood calcium levels are too low, one option of blood calcium levels are too high, and then draw out a flow chart for what happens in each of those situations. So that's a really helpful study tool. This isn't, like I said, the best diagram to explain it. I think there's clearer ways to kind of illustrate it. And if you can create a really clear illustration, it'll be a little bit easier to understand. Any questions about this? Or any bone tissue stuff? Because this kind of wraps up the bone tissue lecture. No? Okay. We are done with bone tissue. So that'll take us into the skeleton on Friday. Start learning some bones on Friday. And we obviously have some extra time. So I actually want to talk a little bit about the next step in your group video project group video project. Yeah. So the actual video part. So let me bring this up real quick. There we go. So you guys on Zoom should see the details for the presentation. Okay, so I have really briefly looked through the outlines, and I think in general they look really good. I think you guys did a great job at getting kind of an initial summary outline together for your project. So good job on that. I will be looking at them and I'll have them graded by the end of the week. And I'll have some comments for you too. So just some areas you can maybe improve or add some more detail. And that's really the next step that you'll need to take. So the next thing you're doing, due in 23 days, March 26th, is the actual video presentation of your disease. So I say you'll record it using Zoom. If you want to use something else, you can. I just always use Zoom nowadays, so it's up to you. I don't care what platform you use. You're just going to post a link to your recorded video on YouTube. And then you'll post that in the discussion board. I'll talk more about that as we get a little bit closer in Canvas, so everyone can actually go watch your video. So you're all going to watch each other's videos and do like a basic summary. That's a little bit further down the line, so not due until like later in April. So I'll talk about that later, but this next piece is kind of the big piece of the project, right? So all of this information is, you've essentially outlined your entire presentation already with the outline you just submitted. So all of this information should be in your video. And essentially, I think all of it, but the works cited was included and required for your outline. So you have all of the information. The next step is to add some detail to that. So the outline is probably a good scaffold for what you have. There's probably a lot more information that maybe you didn't put in there. So you're gonna wanna kind of beef it up a little bit, whatever sections you took care of. So I think everyone kind of divided up the sections. So keep focusing on that section, add some more detail to it, and start to put together the slides. So you'll have a slide presentation, PowerPoint, Google Slides, I don't care, along with your audio presentation, obviously. So you want some slides. I'm going to show you some examples, a couple of examples of videos that were done for another class, for an infectious disease class. So you at least have an idea of kind of what it looks like. I think it's always helpful to see a couple of examples. They're not perfect. They're a really good kind of baseline to start off with. Oh yeah, so your presentation, remember, should be about 8 to 10 minutes, which with 3 people shouldn't be too difficult. And there should be enough information on all of these diseases to at least get to that 8-minute minimum. You can work on them totally separately. So have your own slides, work on your own script, whatever you want to say. And then at the end, kind of bring them all together and make it look uniform. So obviously use the same PowerPoint theme and background, all that good stuff. So yeah, I think the best thing to do right now is just to look at a few pieces of a couple of different videos. Again, these were individually done videos. They were not group videos. So you guys have kind of a benefit. You didn't have to look up all of the information, but you'll have to think about timing. So each person should talk for about the same amount of time. One person shouldn't hog the entire 10 minutes. So definitely work on timing and know who's going when. Right here.
name is Michael McGuire, and I chose to do my disease presentation over Lyme disease. Lyme disease is most commonly caused by the bacterium Borrelia burgdorferi. The first thing I'm going to talk about is the shape and structure of this bacteria.
So I'm going to pause this every once in a while and give you some tips and hints for presentations. So how many people have made a presentation before? Like a PowerPoint. So most people. It's definitely an art. So you get better at it the more you do it. So some tips that I have, not to rip on this poor guy, but some suggestions, I should say. I would cut down on the text on your slides. So as much as you don't think this, you want the audience actually listening to you. You don't want them reading sentences on your slides. So use your slides kind of as a scaffold or an outline, kind of like you already did, to remind yourself of what you're going to say. Hopefully you guys can hear me okay. So try to cut down on the words. You don't need complete sentences. And also here, you'll see he has in-text citations, CDC 2019. I would say don't do that. You can if you want to, if you feel very strongly about it, but I think it just adds, it kind of makes the slides a little bit wordier than they need to be and takes away from the information and from people listening to you. So your works cited will just be a list, like one slide, or if you need more than one slide at the very end, you'll just have a list of the sources that you used. So you don't need to do in-text citations. Okay, so we'll continue on with this.
These bacteria are highly motile and have a spiral shape. In addition, their cell wall consists of a cytoplasmic membrane surrounded by peptoglycan and flagella, and then by a loosely associated outer membrane. The most common way these bacteria come into contact with humans is through the bite of Ixodes scapularis, more commonly known as the deer tick. In this slide, you can see the life cycle of the deer tick. First, the adult females drop off hosts to lay eggs.
And I'm going to stop it here and just say having figures and diagrams is critical. So it's really going to be helpful for you to have, I mean, you know, it won't be a life cycle because it's not, I don't think anyone has an infectious disease, but something similar to use illustrations rather than just all words. If you see PowerPoint slides, I'm sure you probably see some of mine and you're like, oh, there's so many words. You don't want all words. You want some kind of visual interest on your slides. You can have some bullet points next to this if you want, but figures, graphs, and images, make sure you have those on most of your slides. They don't have to be on all of them. You can have some slides which are just text heavy.
Eggs then hatch into six-legged larvae.
Okay.
The larvae attach to and feed on the first host and may acquire the bacteria disease at an early enough time.
So I'm jumping forward here.
The majority of individuals that contract Lyme disease completely recover and experience minor symptoms such as fever, chills, headache.
So even if it's something small, like some pictures to illustrate what you're talking about, definitely put those up there. So like a slide like this, it's just a lot of text, and yeah, including complete quotes is really not necessary for this type of project. But you can also see another note, you can write out a script if you want. So if you're worried about not having enough text on a slide because you think you're going to forget what to say, have a script written out. He doesn't have his image up there when he's presenting. That's totally fine. You don't have to. You don't have to have your face in the corner if you don't want to. So you can be reading off of a script if you want or not. It doesn't really matter. You're not in front of an audience, so it makes it a little bit easier. I will say, don't read like a robot. You have to make it, add some animation so it's a little bit interesting, a little bit of intonation up and down to keep the audience engaged. Otherwise, it's going to get very boring very fast. Okay. So it sounds like he has a script that he's reading off of, but it doesn't sound robotic, right?
Unidentified late-stage Lyme disease can lead to excessive healthcare costs. Many studies suggest that Lyme disease imposes an economic...
So even that little bit of intonation helps, just something rather than this is my disease. It is Lyme disease. I am going to talk about Lyme disease, something boring like that. So try to make it a little bit interesting, act excited about it, even if you're not excited. And as far as the level of detail, imagine the audience is someone who's in college interested in human health, but has not yet taken A&P. So they don't know most of the terminology. And a lot of your classmates won't know a lot of the terminology that you're using because it might be very specific to your disease. So define anything that you think needs to be defined that you were like, what is this? when you were reading through it. Explain that. That'll definitely take up some extra time. So if you're having trouble kind of fitting enough information in to fill 8 to 10 minutes, that's a good way to go. And these are shorter, notice, because it's only one person. I'm going to share with you the Epstein-Barr virus. And you guys on Zoom probably can't hear her very well. So she doesn't have great audio. That's fine. If I'm not requiring you to have like a studio set up, if you don't have great audio, don't stress about it too much. Just make sure you can mostly, like the people listening to it can mostly hear it. So here's just another quick snippet of another presentation. By 3 British scientists, Epstein-Barr and Stone. They found the viral particles in lymphatic cancer cells. EBV is one of the most common human viruses. It affects about 90% of the human population at one time or another within their life. EBV is actually one of the 8 human herpes viruses. It's known as herpes virus 4. The most common name you've probably heard is called cystic disease, and it's otherwise known as infectious mononucleosis. EBV is a double-stranded DNA virus. So here she has text and then a figure, right? And I don't remember if she actually explains the figure, but if you have a complicated figure like this, definitely reference it and kind of explain it. You don't have to explain it in detail, but if it's there, it should be adding to your presentation. And my last suggestion, we'll stop there, but that hopefully gave you a little bit of an idea of what it should look like and what I'm looking for. But other than that, it's fairly open-ended. As long as you include the information is paramount. So good information is the most important part. After that, focus on making it an interesting and clear presentation. And then my last suggestion before you actually, so this is due March 26th by the end of the day by 11:59. Before you run through it, at least time yourself and have your group members time themselves so you know if you're within that 8 to 10 minute limit. And then run through it once before you record it, because you're going to end up messing up. So even if you think we're going to get this done all in one shoot, go ahead and just expect to do one run through, see how it goes, and then a second one, and hopefully that one will be the one that's good enough to submit. If you have some like stumbling over your words a little bit here and there, you don't have to re-record the whole thing. So like I said, it doesn't have to be perfect. Just make sure it's within the time limits, has good information, and is generally a nicely laid out presentation. If you want me to look at your slides ahead of time, I'm happy to. Even if I don't look at every individuals in the group, I can look at 1 or 2 people's and give you an idea if you're on the right track. But I'll have comments back to you on your outlines, which will hopefully help you get started on putting this presentation together. Any questions about that? All right, that's it for today, and I'll see you guys either in lab today or on Friday.